Mitosis is a process of cell division, and it's how all of the cells in your body were formed. Meiosis is a similar process, but instead of making cells to be other parts of your body, it makes eggs and sperm. You can remember this by remembering that meiosis made me, as in it's a process by which you were originally formed. But today, we will talk about mitosis. You can remember the stages of mitosis by remembering I propose men are toads. This might help you in many parts of your life. And it stands for interphase, I, prophase, propose, metaphase, men, anaphase, R, telophase, toads. First, we have interphase. Interphase is the part of the cell cycle in which the cell spends most of its time. Here we see a chromatid, or two identical chromosomes, talking to a tangle of DNA, saying, "Dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet." During this part of the cell cycle, the cell grows and undergoes synthesis, where the DNA is replicated. The second phase is prophase. What divides and multiplies at the same time? Mitosis. During this part of the cell cycle, the DNA condenses into chromatids, which, like I said, are the two identical chromosomes joined by a centromere. The third part of the cell cycle is metaphase. Here we see a cell dividing, and one cell says to the other, "It's not you, it's me." During this part of the cell cycle, the chromatids align along the midline of the cell in preparation for division into two parts. Finally, we have anaphase, which is my favorite part of the cell cycle. And here we have a professor, like me or Miss Garcia, explaining to the students and saying, "And that concludes today's lecture demonstration of mitosis." And as you can see, the professor has divided into two. During this part of the cell cycle, the chromatids separate at the centromere and move to opposite sides of the cell. The final phase of the cell cycle, before fully completing division and cytokinesis, is telophase. So I found this on a website of nerdy pickup lines, and it says, "You're like telophase, I admire your cleavage," which should give you a clue as to what is going on during telophase. During this phase, the cell begins to cleave. The daughter nuclei are formed. As you remember, when the cell was getting ready to undergo mitosis, the nuclear envelope dissolved, and scientists are not entirely sure how that process happens. But we do know that during telophase, the nuclear envelope reforms. Finally, the cell cycle is complete, and the cell undergoes cytokinesis. So here we have a cell undergoing cytokinesis, and one cell says to the other, "After this mitosis, I keep the bachelor pad, the car, and this furniture." And the other cell says, "Well, good luck. I'm calling my attorney." This is where the division of cytoplasm happens and creates two daughter cells. This process actually started back in anaphase and continued on through telophase until finally completing separation. Mitosis is not an equal opportunity process. You have many parts of your body, for example, the brain, the gut, the muscle, the heart, skin, and the liver, and these organs do not undergo mitosis at the same rate. That is to say that the cells inside of these organs do not get replaced equally. And this might make sense to you. One that often makes sense to people as a fast replaced part of the body is skin. If you think about how much you put that poor organ through, it makes sense. It needs to be replaced fairly rapidly. And it is true, it's replaced about once every 2 weeks. If you get a cut, that cut might heal more quickly, but the time it takes for all of your skin cells to turn over is about 2 weeks. The gut is even faster, and think about what some of you put your poor guts through with the type of food you eat. This might make sense. The gut actually turns over in about 5 days. The liver which you may be familiar with because it's important in detoxification of the blood, turns over in about 1 year. If you know anyone who's extremely alcoholic, they can actually damage your liver so much that they are no longer able to have the 1 year turnover rate be a fast enough process for the type of detoxification that they are doing. You guys are getting to the age now where your muscle cells have just about turned over, because those turn over entirely about every 15 years. The heart and the brain are very important, but yet they are not very often replaced. The heart is replaced less than 1% per year, and so you can live out your whole life without entirely replacing all the cells in your heart, and the brain is replaced virtually never. That is why it is very important to avoid damaging the cells in your brain, because you don't get a chance to replace those for the most part. How do we know this? Well, check this out. This, of course, is an atomic bomb. In 1963, there were a lot of testings of these types of bombs, and that caused a lot of carbon-14 to be released. There is a certain amount of carbon-14 present in any case, and you might be familiar with it because it's used to test animal bones and dinosaur bones from long ago. Because you get the carbon that makes up your body from eating food, and there's some amount of carbon-14 present in the environment, you can test how old an animal is, because once it dies, it stops taking in new carbon-14, and so you can just measure the radioactive decay. In a living human being, however, the carbon-14, amount of carbon-14 in the cell can show the age if there is enough carbon-14 in the environment at moments when these cells were made. In this case, any cell that was made in 1963 would have a lot of carbon-14, but cells that were made later would not have so much, and so you can test how much turnover there is in cells in different parts of the body. This work was done by Dr. Friesen from the Karolinska Institute, which is in Russia. Sometimes, however, mitosis goes wrong. There's a disease that you are all familiar with, which can be considered a disease of mitosis, and it is not crabs. We can talk about crabs some other time if you guys are interested. It is, in fact, cancer. Because cancer is essentially a problem in which your cell cycle goes out of control, and you have a vast amount of growth turning into a tumor. The cell cycle is regulated by proteins. What determines what proteins you have? DNA, of course. You can have a process called non-disjunction, which is a very important process, and we'll talk about it again in meiosis, because it can cause very important types of disorders in embryos and in the babies that are born. Basically, non-disjunction is when you are undergoing the division of a cell and the two chromosomes in the chromatid do not separate. So both chromosomes go into the same cell, whether that is a daughter cell in mitosis, which is to say another cell of your body, or whether it's a gamete, like sperm or egg, in which there are 4 cells resulting. What phase does this happen in? Well, as you can see, the cell chromatids are being pulled apart, and that happens in anaphase. There are several different types of ways that you can have non-disjunction. You can have non-disjunction where the kinetochore, or the pulley device, is not attached to a chromosome, or you can have it attached to a kinetochore on either side, so a chromosome is being pulled in 2 directions. You can have a misorganization of the kinetochores where one side has unequal pulling power. All of these can result in your cell winding up with too many or too few chromosomes. This is called aneuploidy, which is an abnormal number of chromosomes. If the added or missing chromosomes contain the DNA that code for the proteins that regulate the cell cycle, what will happen? Well, you'll have division out of control.